This is a brief presentation to summarise our paper, A Phylogeny of Southern Hemisphere Whelks and Concordance of the Fossil Record. The whelks referred to in the title are classified within the family Buccinulidae within the clade near Gastropoda. Whelks are found worldwide, but there's a long-standing hypothesis in molluscan taxonomy that whelks found in the Southern Hemisphere represent an independent evolutionary radiation from lineages in the Northern Hemisphere. Northern Hemisphere whelks are classified as the family Buccinulidae, whereas Southern Hemisphere whelks are classified within the family Buccinulidae. The Buccinulidae clade in the Southern Hemisphere is dominated by taxa from New Zealand. So we wanted to investigate the potential difference between Northern and Southern Hemisphere whelks using molecular phylogenetics. The majority of our DNA sequence data came from high-throughput DNA sequencing. This was used to yield mitochondrial genomic and nuclear 45S RNA cassette sequence data. It was also complemented with short-length sequence data for COX-1 and 16S RNA genes from the mitochondrial genome. So our molecular phylogenetic results indicated that Buccinulidae whelks are not monophyletic. Northern and Southern Hemisphere whelk taxa could not be distinguished from one another based on our mitochondrial and nuclear ribosomal sequence data. The tree presented here is based on mitochondrial data, and the taxa shown in different colours represent putative Buccinulidae from the Southern Hemisphere, whereas black lineages represent putative Buccinulidae from the Northern Hemisphere, as well as Zareie-Vaux and Fasciolaridae outgroups. Our molecular phylogenetic results also indicate that New Zealand whelks are not monophyletic as well. This result is important because the proposed Buccinulidae clade in the Southern Hemisphere was dominated by New Zealand taxa. New Zealand taxa do exhibit high levels of species endemism, but there does not seem to be a single common ancestor within New Zealand for all of these different sampled genera. So how did this result compare to the fossil record? Our molecular trees show close concordance with the fossil record for three putative Buccinulidae genera, Penaion, Caletia, and Antarctineptenia. In fact, the divergent states estimated from our molecular data often matched very closely with the known fossil record for each genus. This molecular result and the close concordance of the fossil record is a really important finding. If you consider the extent and past distributions for these three genera compared to the molecular divergent states and fossil record, it seems inevitable that long-distance dispersal must have occurred in this clade. Therefore, an assumption of biogeographic isolation for New Zealand or the Southern Hemisphere is likely to be incorrect. This means that the Buccinulidae hypothesis is relying on something that is demonstrated not to be true, at least in this clade. To briefly summarise some of the key findings of our study, Southern and Northern Hemisphere whelks are not separate, neither are New Zealand taxa, despite their high species endemism. Buccinulidae is not a monophyletic group, and for three putative Buccinulidae genera, Penaion, Caletia, and Antarctineptenia, there is close concordance between molecular trees that were fossil calibrated using independent dates and their fossil record, and the evolution of the group implies long-distance dispersal, which inherently challenges the idea of biogeographic isolation for the proposed Buccinulidae clade.